talk today is the structure-guided approaches to eliminate PXR-mediated drug depletion. And I want to tell you a little journey that we have taken in GlaxoSmithKline with respect to how we can come up with a rational way of eliminating what we call a severe problem in the pharmaceutical industry, that is PXR-mediated drug-drug interactions. So just a quick outline. I'll give you a little bit of a overview on drug-drug interactions, how serious of a problem that is. But most of the talk will focus on the PXR issue, particularly where the pregnant X receptor, the structure mechanism, what are the differences between the dose and induction. And a bulk of my presentation would be directed towards the medicinal chemistry audience, where what are some of the strategies that we have developed in combination with the what was reported in the literature prior to us getting into this space, and how we utilize this information to overcome on a PXR problem on a chemical series that is very relevant. And finally, I want to close with what are some of the applications, how broad of this the insights that we have developed in GSK could be applicable to move the needle from phenotypic way of overcoming PXR induction to more of a structurally rational way of overcoming the PXR induction. And that's where I want to conclude. Just to build this target, just to give you a little bit of overview, as you all know, this is not a concept that is novice to anybody. Drug-drug interactions can cause serious toxicity and loss of efficacy, and led to withdrawal of several drugs. This is more prevalent in 1990s, early 2000s, less prevalent because pharmaceutical industry have gotten smarter over the years. But nevertheless, 3% of all the hospital admissions over persons 50 years or older, because most of the subjects that are over 50% happen to take multiple medicines, and that's where the drug-drug interactions become serious. And it is costing the industry about 1.3 billion. This is an outdated number, and that figure has gone up now in more recent times. And one of the biggest things that drug-drug interactions is the impacts the companies in 4 different ways. Obviously, revenue, if it happens to be a late-stage drug-drug interactor offender. And obviously, if there is any adverse events that leads to fatality or severe hospital administration, that could impact the company's reputation. Obviously, if it is a fatal things, people will be required by FDA to pull the medicine back, and that could lead to depleted pipeline. And most of it, you don't want to find out you have a drug-drug interaction in the clinic, because it's going to be impacting the cost of development and overall portfolio cost to the pharmaceutical industry. So there are quite a few mechanisms, and today there's pharmacological mechanisms with respect to drug-drug interactions, how one entity would exaggerate the pharmacological effects. I'm not going to focus that today. I'm going to focus rather on the entities that impact the pharmacokinetic properties, particularly driven by drug metabolism. As you all know, CYP-mediated monooxygenases are one of the key drug metabolizing enzymes, and there are 2C9, 2D6, et cetera. But among them, CYP3A4, not so much AFI because it did not come to the limelight as much as CYP3A4. 3A4 is the biggest offender, and greater than 50% of the pharmaceutical molecules have been known to be metabolized by, to some degree, 3A4. So within this, there's, you could inhibit the 3A4 that leads to extended pharmacology and pharmacokinetics. You don't want that. But the biggest one is if it's inducing higher expression of 3A4 that cuts into the pharmacokinetics of the molecule of interest, and that leads to the subactive pharmacology, and basically patients don't see the efficacy, and that could lead to as serious adverse event as death, right? Because if the molecule is not working and you degraded it by induction of PXR, and you would lead to inefficacy of the molecule. And In this context, I'm going to focus on PXR, but there are other nuclear receptors such as aryl hydrocarbon receptor and CAR. They're also known to induce various CYP enzymes, but today my focus is on PXR. And where we found out, interestingly, about PXR is, one of the key projects that I was leading has a GSK compound 817. It's a thiophenol pyrimidinone, thiophenol pyrimidinones at C5 position. And this is a beautiful molecule, showed all the desirable pharmacology we wanted to see. But when we took it into the toxicology studies, we took it into mouse tox studies for 10 days. We dose every day, as you can see in the table. And when we looked at the exposures in 10 days, and we did the 3 different doses, 300, 1,000, and 2,000 mg per kg, especially as we increase the dose and if you compare the exposures from day 1 to day 10, you could clearly see the exposures being a lot less. Instead of going up, they're significantly less. If you look at the 2,000 mg per kg, we're almost 1/10 of the doses compared to day 1. And we initially scratched our heads, but until we got the overall histopathology data, it was very, very clear that we are inducing PXR. We looked at the message RNA. We did the expression. Also, we did the expression of various CYP enzymes. Interestingly, going into this, we knew this is a 3F4 substrate. But now it's not only a substrate, but it's also inducing the expression of the 3F4 via PXR induction. And I'm telling you in a second how they are related. So what this molecule is doing, not only it's a substrate, but induces the expression of 3F4. So it's completely depleting the exposures at day 10, no matter which dose we have dosed the mouse at. So this is obviously a showstopper for this molecule, but we took a step back and surveyed the literature. And interestingly, there's not much help in the literature with respect to rational way of drug designing. What people have done is you pour down all your collection of your compounds in that program through the various phenotypic assays, cell-based assays, and you identify, okay, this set of compounds do not have any PXR. Okay, let's move them forward. Right, which is very trial and error type of approach. So what we have taken is there a rational way of overcoming PXR? So before I get to that, let me give a very brief introduction on PXR. PXR is a nuclear receptor. It has multiple domains. 2 domains of interest is DNA binding domain. This is the domain that binds to DNA and does the transcription of various genes. And this is the ligand binding domain, which is E. This is where the small molecule bind. So if I could draw your attention to this, the DNA binding domain is highly conserved across the species. But interestingly, ligand binding domain is not very conserved. And this is one of the reasons early on in the pharmaceutical industry, they're not seeing the PXR induction in, let's say, in rodent species or dog or monkey. But as we go into humans, because of this ligand binding and compounds are binding in this region, leading to the PXR induction, leading to adverse events or lack of efficacy. So that is a culprit. So, how can we eliminate this? So again, one more step, the way it does PXR is very smart, has evolved because, body has to protect itself from xenobiotics, right? When the molecule enters the hepatocyte and it binds to the PXR, and once it binds to the PXR changes the conformation, it recruits RXR, and it binds this heterodimer, PXR-RXR heterodimer, and it binds to DNA through the DNA binding domain, and induces the expression of 3F4 and 3F5. These are phase 1 metabolizing enzymes. But it also induces conjugation enzymes, glucuronidation, sulfonation, et cetera, and phase 2 enzymes. And it also induces expression of phase 3 such as efflux, right? So basically, the PXR is highly evolved where you could take the xenobiotic, metabolizes the hydroxyl groups, and it conjugates and eliminates through efflux mechanisms. So very highly evolved mechanism to keep the body from the toxic xenobiotics. Beautiful mechanism. And there are quite a few compounds out there that are PXR inducers, rifampicin, bemepizine and St. John's wort. I'm not going to go into that, but typically people use rifampicin as the most potent activator and compare as you find the new compounds that you identify in your programs, how effective is this compared to rifampicin? And that's what they compare it to. And typically, and maybe I'll skip this slide and go to this chart, typically if it is greater than 70% as compared to rifampicin, it's high inducers. If it's somewhere between 30 to 70%, it's possible. So this type of inducers, these are dose dependent. And the other ones is something like this type of a graph. Even at higher concentrations, you won't see the induction as compared to rifampicin greater than 30%. Then this is the box that you typically want to be in if you wanted to have a clean profile in clinical trials. And so we took a rational approach and we looked at the survey of the literature, and there are crystal structures reported, but unfortunately what the crystal structures, even these nice papers in Science and Nature and other respectable journals, what they determined is, small molecules like this, they bind to PXR in multiple binding orientations, right? All of a sudden, as a medicinal chemist, you would be scratching your head in terms of, okay, how can I use this crystal structure if they're binding into the multiple binding modes? But what we determined is if you co-crystal structure with a SRC, which is a cofactor, you may not see these multiple binding modes. Of course, it depends on the structures, but we did not know at that time very early on. So that's what we did. We took our molecules and we co-crystallized with the PXR with the SRC cofactor. And interestingly, what we noticed is, we saw the multiple binding modes in the absence of SRC cofactor, but in presence of the SRC cofactor, we've seen only one binding mode. And this is the co-crystal structure. And it, at the bottom of the pocket, you have two hydrophobic residues, phenylalanine 288 and tryptophan 299. And on the right-hand side, you have a leucine. And on the left-hand side, you have phenylalanine 281. But there are two key hydrogen bonding acceptor donor, that is histidine 407 and glutamine 285. And when we surveyed the literature, these are some of the key key residues from the PXR protein that are shaping up the pocket and interacting with the small molecule that are crystallizing. And this is a very, very insightful insight that we would determine based on not only our observations, but the survey of the literature. And when we noticed with various structurally different rifampicin, those are the same common residues that are surrounding that pocket, and we know that from again survey of the literature. So and we crystallized a few more things, and also they also exist in the same binding mode, but also when we overlaid it with the docking, we have the same binding mode. So we started thinking, okay, we don't need to co-crystallize every molecule in this series. We could use the overlay and docking aspects other ways to understand the SAR of the PXR induction. And one more final thing before I go into the compound-based. And we also looked at various other things, and we saw the similar binding modes as for each one of them. And sorry, I have to say similar residues that they are interacting irrespective of the chemical type. And so armed with that information, we started introducing polar groups where you have the place of this hydrophobic. And let me skip this for the sake of time. And if you see this hydrophobic, and even if it is a hydrophobic, if they are rigid enough, there is no flexible linker, they bump against this tryptophan 299 and will reduce the PXR, which is a 59% compared to 158% as related to rifampicin. So what we thought, if we have put a polar group, it's the same thing, 42% compared to 158%. So what we thought is, can we compare, can we combine polar groups here with the hydrophobic group, and would that be even better? So that's exactly what we did. We put a pyridal group, which we needed for not only activity against the calcium receptor, which is the program we're in, but we put a heteroatom group, which is a pyridine, and it's not only bumps into this very rigid pocket at the bottom of the PXR pocket, but also the polar groups completely removes the PXR much less than 30%, even at the higher concentration. So this falls in our third bucket to being very clean. And this is the compound that we went forward with into the post the candidate selection stage in GSK. Couple of quick observations. We looked at the thousands of compounds reported through the correlations. The C-log-P does not correlate very well unless you are less than 2%, 2 C-log-P of 2, which is not a key attribute. And we looked at various phys-chem properties. Two things that stood out: introduction of rigidity at the bottom portion of the pocket, and introduction of peripheral polar groups, no matter even at the bottom of the pocket, top of the pocket, or left or south of the pocket that I mentioned to you, those making a profound impact on this. And we did what we call built a pharmacophore model. And again, these are the key residues that I mentioned. Again, at the bottom is tryptophan, phenylalanine, and leucine, and the serine. But the two key hydrophobic residues are histidine and glutamine. And you could see the visual in it differently. It's almost like a triangular series. And if you have polar groups, they typically link with the histidine and glutamine. If they're hydrophobic groups, they are interacting with these through. And we recently, just a few months ago, we published this paper. Quite a few details that I won't able to cover here due to the time constraints, but it is available in that paper. Feel free to reach out if you have any questions. But overall, just want to conclude, DDIs is still a serious problem, but I think we have the rational way of eliminating it preclinically using the crystal structures or the docking avenues if the crystal structures become expensive. And also the models that we built in is reported people who are models elsewhere that reported maybe speaks to the core of it, but not as accurate. But at least it'll give you a start in terms of eliminating rationally the PXR induction that seemed to be a big problem. So I'm going to pause there and take any questions you may have.